Well, hey AP, hope you're doing well when you watch this part 2 video of chapter 5. And so let's go ahead and get started. This is a practice FRQ question. Don't look up the answers. It doesn't help you. Try to graph the data on the note packet that I gave you. We'll go through it together, not for a grade, just for practice. Have fun with that, okay? So objectives for number 2 is all about diffusion, osmosis, and the role proteins play in moving, and also hypotonic, isotonic, and hypertonic solutions, okay? Very important. So let's go ahead and talk about membranes. So membranes are really special because they selectively allow certain substances in and out. Two processes deal with this: passive, which does not require energy, and active, which does require energy. So if you actively, well, that's not a really good circle there, is it? But if you actively, I don't know if I can do that. Oh, there we go. If you, oh, sorry, waste of time. If you actively study, or if you actively practice, or if you actively really do anything in your life, you're involving a lot of energy. You're devoting a lot of time and energy to that, okay? And so in biology, I like to think of it like this. There's uphill reactions. Wow, no. There's uphill reactions, and there's downhill reactions, okay? And so uphill reactions are active transport because they require energy, and downhill reactions are passive transport because they don't require as much energy, only a little activation energy. And so simple passive transport happens in a lot of different ways. You can have it simple, which is just straight through the membrane, or facilitated, which means it's helped by what? By proteins, okay? Good old proteins. And so that's important there. So hydrophobic molecules can dissolve and cross easily. Polar ones, this is very important. Circle this, and obviously it's a blank. But polar molecules do not... I messed up. I should have just drawn a box. I'm sorry. I'm trying to get better at this while actively, I should do this before. Oh, that's so much better. Polar molecules, such as sugars, do not cross the membrane easily. You got to know that. You got to know what characteristics of a molecule does not allow it to cross the membrane. So polar is the first thing. Charge is the first thing. Also involving with this is you can also deal with shape as well. Okay? So like larger molecules can't cross very easily, can't cross easily. But if I had to pick one, the most important determination is charge. Okay? So charge is the most important determination. Okay? Transport proteins allow the passage of hydrophilic substances. So some transport proteins are called channels, which means they're always open, and it involves a hydrophilic channel. Wow, don't do that. That's an erase. It involves a hydrophilic channel, which allows molecules and ions to use as a tunnel. Hey, aquaporins, aqua means water. Water's charged, right? Water's polar, so water needs help getting across the membrane too. Other transport proteins called carriers bind to it and change shape to allow it to transport. So here's some questions I want you to answer. You can go ahead and pause this and answer those questions. Diffusion is the process of random movement. Okay, so if you drop ink and eventually over time it goes from greater concentration to less concentration, spreads out. I don't know if you've ever been in like a middle school or any hallway where Axe is being sprayed, but yeah, it can be sprayed in the corner of your eye and then all of a sudden you smell it, right? Diffusion. And so diffusion, this is straight through. This is facilitate, which means it's helped. Active transport is going from low to high. Oh, man. Man. Oh, that's cool, a speech bubble. It's going from low to high concentrate. So that is what's happening there, okay? So, that was interesting. Speed of diffusion, this is a very important slide. I would highlight it. Make sure you highlight the notes and stuff like that. But this is very important. So diameter, small molecules go faster. Temperature, higher temperatures allow the faster diffusion. And concentration grade. All three of these things are very important when it comes to how fast diffusion happens. Some molecules cross by simple diffusion. So gases that are small, look at it, look at the qualities, right? Small, nonpolar, lipid soluble. Polar molecules do not pass. So amino acids, sugars, ions, even water, they cannot pass that hydrophobic layer. Okay? So it's a picture of diffusion. Substances go down their concentration gradient. No energy in passive transport. Osmosis is the diffusion of free water. Water potential, free water, which means it's not bound to anything. And so water diffuses, important, we're going to be doing a lot about it. Water diffuses from a region of lower solute concentration to higher solute concentration. Why? Because there's more free water in a lower solute concentration, and there's less free water in a higher solute concentration because it's dissolving all the solutes. It's attached to it. Water is a universal solute, so it is attached to it. Here's a picture of water. So water, there's more free water here, and then it goes that way and moves that way just due to random osmosis, and eventually you'll have a differing on that, okay? So there's different definitions here you should know. Tonicity, that's fun. Isotonic, hypertonic, and hypotonic. Isotonic is when it's the same inside the cell in the environment. A hypertonic solution is when it's greater outside the cell, so the solution is greater outside the cell, and so the cell loses waters. And then hypotonic is when there's more free water outside the cell than in, so the solid concentration is less outside than inside the cell, so water goes into the cell. And this is how you can actually perish from just drinking way too much water, way too quickly, because you dilute your blood. Water goes into your cells, into your tissues. Your brain can swell. There's been many people, well, not many, but there's been many, documented stories of this happening due to competitions or poor practice when it comes to drinking when you're dehydrated. And yeah, you're losing salts when you sweat, so you need to replace those salts somehow. This is really important when it comes to plants and turgor pressure, because when you have a lot of water, you have turgid pressure, which, if you think of celery and crunchy, celery, why do limp celery stalks crisp up? Because those central vacuoles fill up with water, and it makes them crisp up, right? Flaccid, which means it's very, bendable, and then when it's wilting, that cell is very much struggling with that. Hypertonic or hypotonic environments create osmotic problems for organisms. So osmoregulation is a necessary adaptation for life. So for example, the protist, Paramecium caudatum, which is hypertonic to its pond water environment, has a contractile vacuole that can pump excess water out of the cell. So this is an adaptation allowing it to survive. So here's a picture. So it's hypertonic to its pond water environment. And so water's going to go in, right? And so it needs to pump that water out. Cell walls help maintain water balance. So this is a great picture here of a cell. So there's a cell wall. And so a plant cell in a hypertonic solution swells, and the cell is now turgid. And then if a plant cell and the surrounding are isotonic, there's no net movement, so the plant may wilt, so that's a key word there. And then hypertonic, like let's say salt water invades, or you plant crops, and due to sea level rise, they're now in a salty environment. The membrane actually pulls away from the wall, and usually a lethal effect called plasmolysis. Lysis means to break, and so plasma, of course. So that's like literally cell wall breaking apart. And so you can see movement of water is very important. Turgor pressure is the internal pressure against the cell wall. As it builds up, it prevents more water from leaving. So there is an equation here. Don't worry about that right now. We'll dive into that maybe down the road, practice some problems. I'll walk you through that. Diffusion can be aided by channel proteins. We talked about that. Carrier proteins, both are types of facilitate diffusion. Ion channels. Ions are charged, right? So they're gated. They can be open or closed, ion passage. A gated channel opens when a stimulus, so how do cells respond to stimuli? Well, a stimulus opens and causes the channel to change shape. The stimulus may be a ligand, which I would understand this definition. We're going to talk about ligands all year long. A ligand is a chemical signal. And so we respond to signals, and so that's an important signal too. So here's the ligand. Channel's open, closed. Ligand binds, opens, and now we have some sort of response happening due to that binding. Here is glucose binding to say carrier protein. Glucose is way too big to get in, these proteins to be there. And then here's a scientific skills exercise. I would love for you to do this. Take your time. We'll go over some of this in class. Here's the questions to it right there. Okay? So you can pause. I'm pretty sure it's in your notes, but you can pause this if you need to. God bless, guys. Thank you so much for all your hard work.